A very good morning to one and all. Am I audible? Could you please confirm if I'm audible?
Yes, ma'am, you are audible,
Yes,
ma'am.
ma'am. Yes.
Thank you. So, welcome back to the Ocean Research Knowledge Series, Day 2, organized by Center for Ocean Research and National Facility for Coastal and Marine Research, supported by Ministry of Earth Sciences, Government of India. Today, we have Dr. D. Jayaprakash, Assistant Professor Research, Center for Ocean Research with us to deliver the expert lecture on seagrasses, the hidden forests of the sea in an era of climate change. Welcome, sir. Over to you, sir.
Thank you, Soumya. I'll share the slide now.
Yes, sir.
Hope the slides are visible now, right?
Yes, sir, it is visible.
So, okay. Is that okay, Soumya?
Yes, sir, it's visible. It's entered slideshow mode now. It's visible now. Yeah.
Okay, so do I need to on video camera?
Sir, if your bandwidth is fine, that would be better if you do, sir.
So shall I start?
Yeah, please, sir.
Okay, fine. So good morning, everyone. And on behalf of Center for Ocean Research and National Facility for Coastal and Marine Research, I welcome all the participants for this webinar series. So I'm Dr. D. Jayaprakash, who's working as a scientist and assistant professor research, and my specialization is related with seagrass taxonomy, ecology, and stress biology, which is related with the climate change. So I'm going to handle a topic called seagrasses and hidden forest of the sea in an era of climate change. So I hope the participants are from marine biology background.
We have a mixed participants, sir. All are from life sciences stream.
Okay. So the first thing, so everyone knows, so all the things everyone knows. So apart from that, what I'm going to say is everyone knows that ocean is a blue heart of the earth. So because every second breath we take, right, it depends on the microscopic marine plants called phytoplankton, which releases the oxygen in the atmosphere as like the forest, which is so that we can able to do the breath. And we know that ocean stores more than 90% of the excess heat energy, which is trapped by the greenhouse gases and preventing Earth's atmosphere from warming even faster. The ocean also act as a planet's largest carbon sink, and most of the species have not yet been unexplored in the marine environment. And like 80% of the species remains unexplored, meaning we know more about the surface of the moon than the deep sea. So like that, so still most of the organisms which is existing in the oceanic environments remains unexplored, and this ocean stores enormous amount of heat energy from the atmosphere, and it is playing a major role in mitigating the climate change. So what is the relationship between the ocean and the climate change is ocean is the first and largest responder to climate change. Silently it absorbs the heat energy and carbon dioxide produced by the human activities, and also involved in reducing the involved that in recent years, due to the climate change or due to the geopolitical instabilities. So what is happening is so the carbon dioxide gas concentration is getting higher in the atmosphere, and due to the concentration of greenhouse gases, the earth is getting warmer. So, and because of these reasons, right, we are facing numerous troubles in case of health as well as environmental issues. So all those things we are facing. So considering the marine environment, there are n number of organisms which is playing a critical role in mitigating the climate change. So I'm just majorly going to focus on the habitat foundation ecosystems like corals, as mangroves and seagrasses. Since my specialization is on seagrasses, I'm just going to focus on how the seagrasses are playing a major role towards the climate change mitigation, and what are all the adaptive mechanisms which is undertaken by the seagrasses environment. So in order to reduce the warming or reduce the marine heat waves, so all those things we are going to face. So apart from that, we knew that 2024 was considered as the warmest year of the warmest year event that is recorded globally, and in recent years have continued to rank among the hottest in modern history. So due to that, the organisms which is existing in the marine environment are undergoing vulnerable, threatened, and some in some cases it is lead to an extinction. So all these effects are due to the changes in the weather pattern, changes in the atmospheric gas concentration. So all those things plays a major role in disturbing the marine organisms and the marine ecosystem. So the marine organisms, if it gets disturbed, obviously the organism which is existing in the terrestrial environment will get influenced. So there is a relationship between the terrestrial and the coastal environment. So so the threats, as I said, so what are all the threats is my major threat is the geopolitical instabilities and anthropogenic pressures, which is done by the human activities. So done by the human activities. So all these stuffs are playing a major role in threatening the marine ecosystems and the biodiversity. So mangroves, considering the mangroves, mangrove is one of the is the ecosystem which helps to protect the shorelines from the soil erosion, as well as from so other wave actions, right? So in recent years, there is a report by IUCN that the mangrove ecosystems are under threatened, are considered as a threatened ecosystems, and the habitat which is depending on the mangrove environment, like nearly 1,533 species are are considering the habitat, that is mangrove environment was considered as the habitat for 1,533 species. And in recent years, so all these species, right, so 50% of mangrove associated mammals and other associated animals, rights, all these are under the risk of extinction, threatened and vulnerable situations. So considering coral, as we all know that coral is an animal that is considering polyp, all these things, right? When the temperature rise, so the polyps will get removed from the coral. So due to that, coral bleaching is happening. So all these stuffs are we are going through the records and reports documentations, right, which is related. Even in India, coral bleaching has observed in Gulf of Mannar, Parkway region, Lakshadweep, and Andaman Nicobar Island. So since my specialization is completely related with seagrasses, I'm going to handle in detail about the seagrass environment and how the seagrass environment facing the climate change, acclimatizing it and adapting it to defend the marine heat waves and other stressful environment. So this is what. So seagrasses are considered as the marine angiosperm. So as we all know that it's a flowering plant, so that it occupies nearly three 3 lakhs square kilometer globally. And in recent years, these seagrass ecosystems are getting degraded due to the human activities as well as the weather patterns. So wave actions, currents, all these things. Due to that, the seagrasses ecosystems are getting degraded. Some seagrass ecosystems, like some seagrass species, are acting as a resilient, are making it as a resilient environment, and some seagrass species are considering to be a sensitive environment to the anthropogenic pressures. And 7% of seagrass habitat is being lost as per the report worldwide each year. So considering the seagrasses in India, seagrasses. So considering seagrasses is the is a hidden forest. As I mentioned in the title, it is one of the hidden forest in the sea. So most of the people will confuse with the seagrasses and seaweeds. So seagrasses are the true plant and seaweeds are not true plants. So considering the root system, right? So seagrasses have the root system where seaweeds do not have the root system. So according to that, so most people used to ask seagrasses as seaweeds. So seagrass is a true plant and seaweed is not. So that is what. And in India, the distributions of seagrasses is in Gulf of Manna, Park Bay, Andaman Nicobar Island, Lakshadweep, Gujarat, and Odisha. So among these locations, among these locations, Gulf of Manna and Park Bay host the numerous abundance and diversity of the seagrasses in Tamil Nadu coast. So, and seagrasses comprises two families and seven genera and 16 species. So this is for that is this is a tropical genus. In total, seagrasses have four families, 13 genera, and 16 species. So in India, it comprises two family, seven genera, and 16 species. So all the species are available in India. So and at global level, nearly 60 species are existing. So what seagrass ecosystems plays a major role is seagrass ecosystems is one of the major habitat for the dugong. Dugong is a sea cow, so which and it is vegetarian. So it will that is the food for dugong is only seagrasses. So if seagrasses are not available in the marine environment, obviously the dugong will get extincted. So in the recent report, that is in India, the total population of dugong is nearly 150 to 200 individuals are existing. So obviously, this dugong population is under threatened stage. So if we conserve the seagrasses, right, of the threatened state in future, it may lead to extinction. So if we do focus on the seagrass research and seagrass conservation and restoration, so we can able to protect the dugong population right from the extinction. So that is what Tamil Nadu government have initiated a dugong protection center near Tanjavur. So it is going to get initiated as early as possible. So which is related with the seagrass conservation too. And apart from that, green turtle. So green turtle will provide that is used as the habitat. So apart from other these two, these are the major organism which depends on the seagrasses as a food. So apart from that, numerous organisms like associated fauna, including fishes, gastropods, amphibians. So all those things are existing in the seagrass environment. So as a food and as a that is as a feeding environment, feeding ground as well as a nursery ground for the other existing organisms. So what is the threat which is existing in the seagrasses environment is actually the major threat is anthropogenic pressures, which is also done by the human activities. So during the catching, so using the nets. So when using the net in the seagrass environment, obviously it will the root system of seagrasses, and obviously the degradation occurs. So if the degradation occurs, right, in some cases, if the seeds have not been dispersed, the seagrasses will obviously damaged from that specific place, and in future in scenarios, seagrass environment will not regenerate. So this is one. So as I said before, ocean warming. So how these seagrasses are related with the ocean warming is, so seagrasses is also a plant system. So which focus on that is which is an organisms which also responds to the marine heat waves as well as the climate patterns, even with ocean acidification, ocean warming. So for all these kind of stress factors, so seagrasses will respond. And coming to the research in seagrasses, right, so some species, right, can able to withstand. That is, it is like a pattern wise. The seagrasses depends. For example, a one decade or one for a decade, some species will show a resilient, and the next decade some species will be sensitive to the same climate scenarios. So this is what the pattern which is occurring. So the species which has existed in the past decade, right, their abundance and diverse, that their abundance was reduced, and the other species which that is the abundance of the species, that is the lower abundance of seagrass species got increased. So like Halophila ovalis in the past decades, the population of Halophila ovalis in the Gulf of Mannar and Parkway region was most higher. But in recent days, right, the population is the population has declined in terms of abundance wise. The population of Halophila ovalis got declined in the Gulf of Mannar and Parkway region. So, and rather the other species like Samidacea cerulata, Samidacea rotundata, and the species abundance has increased, and the cover is also getting increased in the Parkway and Gulf of Mannar region. So all these are getting influenced by the climate factors and other anthropogenic pressures. So why seagrasses? So seagrasses, why we have to study seagrasses? Seagrasses is as like mangrove environment. So mangrove environment is like a coastal plant. So when seagrasses are considered as a true marine plant, which is also playing a major role in mitigate, that is in soil erosion, as well as this seagrass environment is playing a major role for protecting n number of organisms in trapping the sediment particles that is n number of photosynthetic organisms in trapping the sediment particles, and it makes the water much shallower in order to perform the photosynthesis process. So apart from that, this seagrass ecosystems are dependent by n number of organisms. So without the seagrass environment, the organisms may migrate to some other ecosystems, or migrate to some other, from tropical to temperate, or else lead to an extinction. So in such cases, right, so we should protect seagrasses, and we should conserve seagrasses and do a science-based research in order to perform the restoration of seagrasses. So in order to protect all the organisms which is depending on the seagrass ecosystem. So these are the species which is existing in India. So among these species, right, the Cymodacea cerulata and Cymodacea rotundata, halodual species are dominant in the Gulf of Mannar and Parkway region. So the first place that is the in abundance wise, Cymodacea cerulata is higher than the other species. So so this is the seagrass species. So these are the distribution maps. So where the seagrass has been distributed. So as like told, Parkway, Gulf of Mannar, Lakshadweep, Gulf of Gujarat, Chilika Lake, and Andaman and Nicobar Islands. So all these places, the seagrass ecosystems are existing. So other than this, these are the major seagrass abundant site. So apart from that, these seagrass ecosystems are also existing in the Pulicat Lake. That is in Pulicat Lake, that is Halophila ovalis species are highly dominant over there. So not only the marine environment, these seagrasses also existing in the brackish water. So but not all the species. Some species which can able to tolerate the salinity, lower salinity, higher salinity, the species which are resilient to that. So it can able to adapt the estuarine environment. So not all the species. So that is nearly 4 to 5 species are existing in the estuarine environments. So these are the distributions. So state specific and site specific seagrass distributions in India. So seagrass distributions in Tamil Nadu. So Tamil Nadu, that is Gulf of Mannar and Parkway region. Gulf of Mannar Biosphere Reserve is considered as the major hotspot of biodiversity, marine biodiversity, and Parkway region too. So so in Gulf of Mannar and Parkway region, the depth of the seagrass where seagrass will exist is nearly 57-meter depth till seagrass can exist so that light can pass in order to perform the photosynthesis. But in the Parkway region, the maximum depth is 13 meters. So these seagrasses will existing from point that is 0.1 meter to nearly 13 meter. So in Gulf of Mannar region, it may exceed to 20 or 25-meter depth. So seagrass will exist. And apart from that, this seagrass environment, this seagrass, in order to perform the photosynthesis, seagrasses will undergo a different kind of stress responsive mechanisms and metabolisms in order to protect themselves, defend themselves towards the climate change. And there are seven genus and 15 species that is existing in Tamil Nadu coast, and the 16th species like Halophila minor is one of the species which are not reported in Tamil Nadu, but it has been reported in Odisha coast and Gujarat coast. So this is what the species which is existing in Tamil Nadu region, and among the dominants, Cymodacea cerulata, Cymodacea rotundata, followed by Halodude, and Enhalus acroides, Thalacia hemprigi. So these are the species which are highly dominant in the Gulf of Mannar and Parkway region. So, uh, so th- so before we perform our research, right? Before we perform research, our main motto is to conserve seagrasses with a science-based research. So in order to, apart from all these research, right? So once the research has been done, the final thing is to take our research into the societal benefit. So for societal benefit, in a sense, obviously the seagrass restoration. So for seagrass restoration, so what are all the science-based research we can perform, and that is what we are doing under Satyabama Institute of Science and Technology. So here at Center for Ocean Research, so we have established halophyte conservation lab and underwater monitoring lab. So all these labs will help us to perform the research which is related with the seagrasses. So halophyte conservation lab is established with the micro propagation chamber as well as the mesocosm chamber, and underwater monitoring lab is established with the scuba diving kits where where the certified divers will undergo scuba diving for monitoring the biodiversity which is existing in the marine environment, that is specifically in the Parkway and Gulf of Mannar region. So other than that, we'll go to Pulicat Lake for doing that is for monitoring the seagrass ecosystems. So what we used to perform here is that is since apart from these that is micropropagation, mesocosm, and doing scuba, right? So all the ecological data will be collected. So the instruments which is existing in Satyabama in Center for Ocean Research, so we have all the ecological instruments which helps us to collect or will helps us to generate the data which is related with the biodiversity monitoring, environmental impact assessment, and how these biodiversity are getting influenced and getting resilient towards the anthropogenic pressures. So all these studies will be carried out at Center for Ocean Research, which is related with the seagrass research. And coming to micropropagation, so I can proudly say that Satyabama Institute of Science and Technology and Center for Ocean Research is having the micropropagation, only the micropropagation of seagrasses, and the specified lab is existing only in Satyabama. So where we have successfully established a callus and cellular suspension from the seagrasses. So apart from that, mesocosm setups are available at our center. So where we can perform the experiment like temperature experiment, acidification experiment, as like the field condition, what are all the parameters which is recorded from the field conditions. So all the parameters can be mimicked in the mesocosm setup, and we can expose the organisms, we can expose the seagrasses to different set of temperatures, different set of pH and salinity, in order to understand what are all the mechanism which is undergoing by the seagrasses to defend themselves against anthropogenic pressures. So all these studies are being studied, all these research are being done at Halophyte Conservation Lab and at Center for Ocean Research, Satyabama Institute of Science and Technology. So when we start our research, right, our main motto is to conserve the seagrasses seagrasses. So for conservation, what we should do is we should focus on the restoration. So if we want to do restoration, as like we can take the plant and in the terrestrial environment, we can take the plant, pluck the plant, and we can plant it somewhere. So it will start to regenerate. But when considering the marine environment, it is not like that. When we take seagrasses and transplant it to some other places, right, it will not, that is, give 100% result. Even the failure has recorded. So in order to do this restoration in an effective manner, a science-based research is necessary to get the 100% success or nearly 90% success for seagrass restoration in India. So that is what our Center for Ocean Research is doing for restoration of seagrasses with science-based research. So our study area is that is from Adramapattinam to that is all the Parkway and Gulf of Mannar region is covered. So since Gulf of Mannar region is a protected area, so in some place sites we will get permission from the forest department and will get access for the monitoring and collecting the samples. But in Parkway region, right, we can perform the research wherever we want. So usually, we used to take the Parkway region for the studies because most of the biodiversity, seagrass biodiversity, are also existing in the Parkway region. So here I have mentioned the donor site and recipient, that is donor site and recipient site. So donor site is nothing but where we can collect the samples in order to perform research, and and the recipient site is like where we can able to do the restoration. So for restoration kind of things, right? So not only the own factor is involved. So for restoration, we should collect the data, n number of data which is related to the physical parameter, chemical parameter, and biological parameters in order to fix or in order to select the site for restoration. So case study. So what research we have done. So this I'm going to update the research which has done so far in our Center for Ocean Research. So related with the seagrass environment and seagrass conservation. So first one is marine heat wave driven seagrass response to seagrass response to climate change. So actually, for a year, we have recorded the data. We have collected the data in the Parkway region. In Parkway region, we have selected the site called Olekuda. So where the seagrass abundance and seagrass diversity is higher as compared to the that is. We selected in order to easy accessibility of the studies, and in order to deploy the instruments to collect and retrieve the data related with the anthropogenic pressures as well as the seagrass biodiversity. So that is why we have selected the sites of Olekuda. So the seagrass environment usually existing in the intertidal region where it will handle the high tide as well as the low tide regions. So for during the high tide, we use that is the divers, which is the divers with the help of scuba diving. So we used to monitor the biodiversity and collect the data. And during the low tide, we can directly enter into the field and collect the data which is related with the physical, chemical, and biological parameters. So the data which we have collected, right? For a year we have collected the thermal data as well as the pH and salinity data. So since we are majorly focusing on the one set of parameters, right? So that is we are working on the parameters like pH, temperature, and salinity. So first we have targeted the temperature. So how these temperature is influencing the seagrass biodiversity and how these fluctuations will make the seagrasses resilient, will make the seagrasses sensitive. So all these studies will be was carried out at our center. So we have recorded the marine heat waves in the study area recorded the marine heat waves in the study area where that is the optimal temperature of the seagrasses is 29 degrees Celsius. It will that is the ranges between 26 to 29 degrees Celsius, but the optimal range is 29 degrees Celsius. And the when the temperature increases, as like even if it is increased to 1 degree Celsius, there will be some changes in every organisms, right, in order to defend to that pressure or to that stress. So the same way, seagrasses also make a defense defensive mechanisms with towards the anthropogenic pressures as well as the temperature rise. So we have recorded marine heat waves during a study period of 2024 to 25. So we have recorded the marine heat waves of temperatures like 35 degrees Celsius and 37 degrees Celsius. So we conducted three set of experiments. So we have collected all the ecological data, including the physical, that is physicals, including temperature, pH, salinity, DO, and all those things. Sorry, temperature, physical, pH, salinity, and chemical DO. All the parameters were done, and the data was collected. And whatever the data which we have collected in the marine environment, right, all the data was mimicked in the mesocosm setup in order to grow the seagrasses under the laboratory conditions. So that is what we have done. So these are some data which was recorded in the field conditions with the help of related with the seagrass research. We are having the instruments like fluorometer in order to understand the photosynthetic pattern of seagrasses towards the temperature or and normal condition as well as in climate change scenarios. So apart from that, we are having infrared gas analyzer, so where we can do studies related with the gas exchange between the atmosphere and water, and that is water from water with the seagrasses. So all these studies will be done with the help of the instruments. So during the studies we have done some experiments like priming and non-priming experiments. So priming is nothing but actually priming, that is control, priming and non-priming. Three set of experiment was done in the mesocosm setup. So control is nothing but whatever the data which we have collected at the field condition was mimicked in the laboratory condition for seagrass growth and studies. And for priming is nothing but that is uh, for example, if 30 degrees Celsius, that is 29 degrees Celsius is control. So if I raise the temperature to 30 degrees Celsius, so what is the mechanism which is changing? And 31 degrees, so each that is at every temperature interval, at 1 degree temperature interval. So what is the changes? What are all the patterns which is existing, which is playing a major role in defending the stress? So all these was studied in the priming experiment. And non-priming experiment is nothing but if an organism is exposed directly to extreme pressure. For example, if an organism is growing at 29 degrees Celsius, if it directly exposed to 41 degrees Celsius, so whether it leads to a lethal condition, or what is the mechanisms which is undergoing in order to defend themselves, in order to uh, protect themselves for the regeneration. So that is what priming and non-priming. Priming is nothing but on interval basis, and non-priming is nothing but from optimal to extreme pressure. So that is what priming and non-priming. So all these experiments were done. And second case study is, so we have done a micropropagation of seagrasses. So micropropagation is nothing but so inducing the callus. So for inducing the callus and cellular suspension. So why we have to do this micropropagation is already micropropagation and plant tissue culture work are existing for the terrestrial plants, and but when compared to terrestrial plant, marine plant, that is seagrasses are the only marine plant so that that is uh, establishing callus and cellular suspension is a risky task for us in order to perform too much research to get the proper results and too much that is chemical compositions, right? We have made a chemical composition and optimized the protocols, right, in order to induce the callus and cellular suspension. So why we have to perform the cellular suspension for seagrasses is already seagrasses are under decline condition due to anthropogenic pressures and other stress factors. Already seagrasses are under decline conditions and at global level. So if I want to work on seagrasses, so being a seagrass researcher, right, my motto is to conserve seagrasses, right? I don't want to distract the seagrasses. So even if I want to perform the research in the field condition, I can able to collect a small amount of samples so I can perform the research. But when I want to study the entire seagrass patterns related with the change, right, I should go and collect the samples. I should go and disturb the natural ecosystem in order to study all these kind of stress experiments. So in order to avoid in disturbing the natural environment, so we turned our research towards the micro propagation side, so that we can, if we establish a callus or cellular suspension of seagrasses, so that we can able to acclimatize the seagrass cells. So whatever experiments we want to perform, right, we can acclimatize the seagrass cells towards it. And also, once establishment of seagrass cells has been done, so it is easier for us to mimic the environment as compared to the mesocosm. So mesocosm is like we have to collect the plant material from the field conditions, and we have to plant it in the laboratory condition, and we have to observe whether the regeneration is occurring or not. So in most cases, and most plants cannot, but we have optimized some protocols in order to regenerate the seagrasses in the laboratory condition. So instead of that, we also using the micropropagation cells, that is cellular suspensions for stress. So these are the callus and cellular suspension, that is, this is the callus which has been established from the seagrasses, and the species is Allodium pinifolia. So we have successfully established the callus and cellular suspension uh, of seagrasses. And for the viability of the cells, we have observed in the microscope. So all these data has been published. So these are the pathways which we have recorded during, that is when it undergoes the controlled conditions, right? Controlled condition, what are all the metabolic pathways it is undergoing, and when it is exposed to the priming experiment, that is temperature rise on interval basis. So what is the pattern, and on the extreme pressure, what is the metabolic pathways it is undergoing. So all these kind of studies were done in order to this is actually for conservation and restoration work. We should understand the complete mechanisms of seagrasses in order to perform the restoration. If not, the restoration process or the restoration which we are going to perform, right, will lead to a failure. So in such cases, right, so understanding the patterns, understanding the mechanisms of seagrasses is really necessary to perform the seagrass restoration. So these papers got published. And case study 3 is that is thermal priming. So this is what we have done. So the prime, that is all the data were collected from marine environment. So, and we have done, this is the mesocosm setup which is indigenously done. So where we can perform the experiment like control priming and non-priming. So as like I said, control is the normal, priming is like interval basis, temperature rise, and non-priming is from optimal to extreme pressure. And this is the image which shows how we have performed the experiment. So accordingly, we have collected the data of fluorescence, that is morphology, anatomy, and physiology data, and also epigenetic data also done related with molecular analysis. So all these observations were made in order to understand the mechanisms of seagrasses towards climate change, that is defensive mechanisms of seagrasses towards climate change. So these are the some of the data which we have performed at our Center for Ocean Research. So since we are having a high end of microscopic lab like molecular imaging hub, as confocal microscopy, micro CTs, right? So we are having enormous facilities in order to perform all these experiments, right? So we should thank Satyabama for providing such facilities, such world-class facilities to perform our works, which is related towards the environmental changes as well as the conservation. And case study 4 is at first we have done the field day collected field data, and second we have done the established the micro propagation, and third we have done the mesocosm experiment, and fourth is seed based seagrass restoration. So seed based seagrass restoration, why we have gone through it. So when we are collecting the plantlet from the field, and we have to perform some optimized protocol in order to carry the plant from field conditions to the laboratory condition. The same thing from laboratory condition to the field condition, right? The same optimized protocol need to be performed, and it is highly risky process, right? If anything need changed, right? The our motto is restoration, so restoration will get in a failure thing. So in order to do right all these parameters, we should focus. So but when compared to a plantlet, right, seed is much easier for us to collect as well as to store, and to carry from field to laboratory, and laboratory to the field conditions. So the experiment, what experiment we have done is the same priming experiment was done for seeds as well as the plantlet. So the same kind of experiment, priming, non-priming, all the things were done. So the seeds were collected during the seeds were collected in the Parkway region, and it was processed in the laboratory conditions. So where we have used Hessian bags in order to do a sustainable restoration. So and sediments, right? What are all the parameters which we have recorded is all physical parameters. So apart from that, what are all the samples collected is we have collected sediment samples, seagrass sample, that is seagrass seeds, as well as the seagrass flowers. So all these samples were collected. So apart from that, we have recorded the data which is related with the site where we are collecting the seeds and where we are collecting the plantlet. So in order to study the restoration site, in order to understand where the restorations can be done. So once the seeds and flowers were collected from the field condition, it was transported to the laboratory conditions. The seeds were processed with the help of help of my scholars, and it was packed in Hessian bag. So Hessian bag is nothing. This is the Hessian bag actually. Before getting Hessian bag, we tried with the jute bag. So that is what the image shows. And we have performed some laboratory condition, that is we have performed some mesocosm experiment with the laboratory condition. That so this is the processing mechanisms of seagrass seeds. So once transported to the lab, in order to remove the detritus and remove the other particles which is attached with the seagrasses and seagrass seeds, right, we have done some process, and we have done a packing with the Hessian bag for restoration. So these are the some microscopic images which is related with the seagrass seeds, that is seed cap, and inside the seed cap seeds are present. So these are the seeds which we have taken for our studies in order to do the regeneration of plantlet from the seeds, and these studies were done in the laboratory conditions. So this is the experiment which we have performed. So in each aquaria tank, the sediments which we have collected in the field conditions was processed, like we have done an autoclave, and we have sterilized the sand materials, and all these experiments will be carried out um, at aseptic conditions. So in order to avoid the contaminations. So these are the results which we got. So once that is in the third week, that is in the first, that is 0th day, we have soaked the seeds in the aquarium tank, and the third week, fourth week, and sixth week, eighth week. So all these results shows the regeneration, a successful regeneration of seagrass we have taken for the priming and non-priming experiment. So these are the measurements which we have made. That is whatever the seagrass plantlet has regenerated from the seeds. So these are the measurements we made. So seagrass restoration workflow. So what we have done is, so in the thermal priming experiment with the seeds and the plantlet, what we have observed is, as like the human beings, right, as like other animals who which is having a stress memory, right, seagrass is also possessing the stress memory, right. For example, if the seagrass is exposed to 30 degrees Celsius, since it is the optimal is 29, if it is exposed to 31 degrees Celsius for a period of time, these seagrasses are storing that I have faced this temperature. So if in future, if it comes, I can able to face. So this is the defensive mechanisms which is played by the seagrasses through the help of stress memory. So not all the species is having the that is stress memory response. The that is some species, that is the fold changes of these stress memories varies between the species to species. So accordingly, the results were done and the paper got published. So finally, the restoration. So all the experiments were done. So we have successfully regenerated the plantlet and successfully generated the experiments, and also induced stress memories in the seeds. So the final thing is to go for restoration. So for doing a restoration, right, we should select the site. In order to select the site, we should perform some we should perform some experiment in a sense. We should perform some remote sensing work, like performing habitat suitability modeling. This habitat suitability modeling is nothing but we should give a data related with the environmental parameters which we have, which where we are going to do a restoration. So all these data has been collected. So and it was given as an input with the help of this tool. So we should develop a map like habitat suitability modeling. So based on the modeling, right, so we will finalize the site where we are going to perform the restoration. So these are the seeds which we have packed in hessian bag and transported to the laboratory. So and the image which is showing here is poti putki. So this is one kind of small minor instrument which helps us to plant the seedlings in the recipient site. So this is what. So these are some restoration protocols which is done along with my scholars. And once done, post plant care, that is once a restoration done, that is we have done a restoration in a small site, like 2.5 square kilometer. So in order to, that is, it is kind of field trial where whether the plantlet, that is whether the seeds, whether the plantlet which is regenerated from the seeds under laboratory condition, the same pattern is reflecting in the field condition. So this is what our study. So, and we have done some trial in the field conditions towards restoration. And after the restoration being done, post plant post planting care were performed. Post planting care is nothing but we have allotted fisher community people that we have performed this that kind of experiments in the field conditions. So we need someone to hold that to monitor, so not to disturb, and we should inform. Actually, we used to conduct the programs to the local communities, local communities. We used to created an awareness which is related with the seagrass conservation. So all these things were done, and long-term monitoring evaluation is also ongoing in the Olekuda region. So where which is a study site. So once actually this is the restoration which we have done. So you can see the site, like the empty site where the only gastropods are existing. So this is the this is the site where we have observed after after the restoration. So after a period of 1 month and 9 days, we have recorded the regeneration of plantlet. So when we have done the abundance, and we have done we have taken the data which is related to the seagrass growth, size measurements, so morphological measurements, all those measurements were made, and observations were done. And also and that is a sea anemone was observed at our restoration site. So this image is reflecting that seagrasses is one of the major ecosystem which is bringing the biodiversity back to ocean. So that is what we thought. so we got a success. Whatever the data we have generated under the laboratory condition, the similar data was reflected in the field condition too. And also all these awareness programs were conducted among the public fisher community, which is related to our research, with the help of our Vice Chancellor, Dr. B. Shilarani ma'am, and our head, Dr. Inbakandan sir. So with their support, we have organized awareness program and Seagrass Conservation Day, Seagrass Day. We used to celebrate at the Rameshwaram coast. So all the fisher communities will that is will gain knowledge towards this awareness program, and they are helping us in order to protecting and conserving the seagrasses in the coastal environment. And these are my research scholars who is being with me and who is there for me, so that all these research were done along with these with my research scholars. So thank you all. Any queries?
Thank you, Dr. Jayaprakash. The forum is now
Yes.
open for queries. Participants, if you do have any queries, you can post them in the chat box or you can directly reach out to Dr. Jayaprakash. Sir, if you could display your contact details.
I don't have. I didn't
Yeah,
mention the contact details here.
yeah. So Dr. Jayaprakash's official mail ID and everything else about him is available in our website, in our official website. So if you do have any queries or you want to reach out to him for internship opportunities or project opportunities, you can do so by checking our official website. And if you could see in the chat box, we have shared few links for the MSc admissions open marine biology and the consultancy charge sheets of Center for Ocean Research and National Facility for Coastal and Marine Research. Center for Ocean Research is open to consultancy in all analytical instrumentation aspects. Just in the chat box, you can find Dr. D. Jayaprakash's contact details also. And, we're getting a few congratulatory messages from the participants. Thank you, Dr. Jayaprakash. Thank you for joining with us today.
Thank you, Sonia.
Yeah.
Couldn't just-
So with this, we come to end of the morning session. In the afternoon session, we have Dr. Ravi, who will be speaking about DNA barcoding and polykit taxonomy. So I request all the audience to join us again at 2 p.m. using the same meeting link for another interesting talk. Thank you all.